Welcome in, everybody. Good morning, good afternoon. We've got an audience joining us from around the world today, and welcome to another exciting broadcast with us here at Exploring by the Seat of Your Pants. I know we've got a lot of familiar faces in the crowd today, but if you are new to us, we are all about bringing conservation, adventure, and science into classrooms around the world. Now, 2023 has been a whirlwind. We have been to, I think, 20 countries on 6 continents to start off the year. It's been crazy. Joe and I just don't sleep at all, but I am particularly excited because we are diving in with this third part of our incredible series for IMPAC5. IMPAC5 is the International Marine Protected Areas Congress happening right now in Vancouver, BC in Canada. It has been the world coming together to work towards a future with healthy oceans. If there's a better cause than that, I don't know what it is, and I'm so thrilled you are all joining us as we've had the chance to showcase some of the coolest young ocean professionals on this planet. Now, the IMPAC5 series of programs has been made possible in conjunction with the awesome teams at the Canadian Ocean Literacy Coalition and the All Atlantic Blue Schools Network. I really hope you get the chance to check them out, and I will put them in our chat in a minute. Before we dive in with our speaker today, I'm going to bring in Diz Lethero. She is the founder, the leader of the Canadian Ocean Literacy Coalition, to explain a little bit about what IMPAC5 is all about before we introduce our star of the show today. So Diz, welcome to the broadcast again, and take us away.
Thanks so much, Jesse, and good morning to all the students, and good afternoon for some of you. It's an absolute pleasure to be here with you today and to be able to introduce Neha. IMPAC5, as Jesse said, brings together global leaders around the world focusing on ocean conservation. So we've got scientists and policymakers, Indigenous leaders, early career professionals, educators, storytellers, all coming together for a common goal. We all know that a healthy ocean is essential for the future health of all of us. And a global ocean, there's only one interconnected global ocean, and so it's a global effort. And we've got to work together to ensure we meet some of the important targets. Protecting 30% of the ocean by 2030 is one of those targets. The other thing I just wanted to share in this opening is that the early career ocean professionals and the people you're getting to meet over these couple days this week, and Neha will be today's expert sharing with you, they have not only shown up, but they've actually played a huge role in organizing this conference, in leading the sessions here and the conversations that we're having. And it's just really important that the young voice is part of these leadership decisions. The innovation, the expertise is essential. And we look at you as part of this. I mean, to have students part of this conversation is really important. So thanks so much for showing up today, and give your teacher a big thanks for making space for this. It's really important. I'm going to turn it back to Jesse and to Neha. Thanks, everyone.
Fantastic. Thanks so much, Diz. And Neha, if you want to bring up your presentation now as I do a little overview of what you do and all about your work, you're welcome to do that. I will note, if you want to learn more about Neha, she's got the coolest website in the world. So check it out below wherever it is on your screen when you're done this broadcast. She joined us first for World Oceans Week, which is in early June last year. And I'm not supposed to be biased. I have to maintain professional detachment, but I've got to say, hers is my favorite program of our series we did in conjunction with Diz and her team back then too, because her focus as a conservationist, a scientist, a professional diver and more is on the understanding of the ecosystem in the ocean. And she does that using one of my very favorite scientific tools in the world, which is eDNA. This is something that's only really come into the forefront in the last decade or so. It provides so many opportunities for exploring wildlife in a way that has never before been possible. And so I'm not going to steal any more of her thunder. I'm going to turn it over to Neha to blow your minds. Thank you so much for being here with us today, and take us away. Welcome to the broadcast.
Thanks, everybody. Okay, so I'm assuming you're just looking at this picture of fish.
Yeah, we're good to go. Seeing the invisible.
Okay, great. Yeah, so today I'm going to be talking about who I am, how I engage with the ocean, space, and some really cool new technologies that we can use to essentially see the invisible in the ocean. I just want to start this talk by acknowledging that I'm sitting here today on the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh people. And I'm really, really thankful that they've been generous enough to allow us to have this really important meeting here. So with that, again, thank you to you, Jesse, Exploring by the Seat of Your Pants, the Canadian Ocean Literacy Coalition, for allowing me to do this live from IMPAC5. I'm very excited to talk to you guys today. This is my second time doing this, but I have done some research since my last time, and so I'm going to be talking to you about some of my new work that I've been doing. So I hope you're excited. Okay, so first of all, who am I? As you guys get older and you figure out who you are, you're going to realize that you don't just fit in one box. You have many different boxes and sort of different people that you are. And I want to talk about who I am in relation to the ocean. So first and foremost, and most importantly, I'm an ocean advocate. I've literally dedicated my life to understanding and working to protect the ocean because we all need it to survive. I'm also a marine scientist, so a lot of this advocacy I do with like literacy work, working with COLC and the early career ocean professionals, but I also love science. I'm a huge nerd, and so I am currently doing my PhD at the University of British Columbia, and I'll be talking more about that research in a bit. I'm also a freediver. I don't know how many of you guys have spent much time swimming or in the water, but if you haven't had a chance to hold your breath and go under the water, I really, really suggest you try it because it feels so good. So freediving is essentially learning how to use your breath and to slow down your heart rate to spend time under the water. And it feels so good, not only just from a body perspective, but also for your mind. It's really good for calming stress and easing anxiety, and also for the soul if you like hanging out with fish, you know? Okay, so I also am a commercial and research diver. So what you see me doing here in that very good-looking suit with that hood there is research diving. And so as a research diver, essentially what I do is I go down and I get to count fish or all the animals that I see, and I love it. I get to go to some really cool places that not a lot of people can go because you really need to have a lot of infrastructure like boats or people who live in remote communities that invite you to come do this work. So I feel so lucky that I'm able to do research diving in these places. And finally, I like to call myself an explorer. So technically, the word explorer means that you visit and you go to new places that not a lot of people get to go to. So I have done that, and I feel really lucky that I've been able to see some really cool places in the world. But I also like to think of exploring as a way of looking at the world around you. So I think all of us are really explorers when we look at the world in a curious way. And I like to also call myself a molecular explorer because I am looking at the ocean through a molecular lens, which I'll talk about in a bit. Okay, so just quickly before I get into the science, I just want to show you some pretty pictures. I've been really lucky to, again, have gone to some really amazing places. So this here is in the Philippines, and I went to the Philippines to learn how to use an underwater camera. You can see me doing that here. And I want to show this picture because what you can see underneath me is a coral reef. And honestly, I didn't know if I was going to be able to see such a healthy coral reef in my lifetime. But I did, and it was amazing. And what's been really cool at these meetings is you're seeing a lot of young people, and I'm sure many of you are going to do this as you get older. They're more focused now on solutions. So there's so many cool talks about like coral restoration and coral gardening. And it's really cool to see that mindset shift from, oh no, everything's dying, to, okay, like how are we going to fix it? Okay, so anyways, I learned how to use my underwater camera. And since then, I've been able to take some really cool photos. So over here, I'm just showing you some animals that I think most people like. So we have some dolphins in Hawaii. Up on the right corner, that is a giant oceanic manta, and that's in the Revillagigedo archipelago, which is the largest marine protected area in North America. And on the bottom, we have an orca. And that was actually in Cabo Pulmo, Mexico, which is the oldest marine protected area in North America. And it actually was established the same year I was born in 1994. And these are some other pretty pictures. These aren't as big, but I think they deserve as much attention. So here we have a loggerhead sea turtle that's taking a nap on the sea floor. And above that is a mimic octopus. I don't know if you guys know, but mimic octopuses can camouflage themselves using color and the shape of their body to mimic 8 different animals. They can look like a sea snake or a manta, or not a manta ray, a stingray. It's crazy. And maybe after this talk, you guys should YouTube mimic octopus because it is really, really cool. And then I just want to remind you guys not to forget about the tiny stuff. So these shrimp here are literally only 2 centimeters long, but they are beautiful, and they are called sexy shrimp because they hang out and they wave their butt in the air as a way to breathe, essentially. So, all right, so these are all in warm water, but most of my work here in Canada is in the cold water. This is actually not in Canada. This is in Antarctica, where I was working with some Chilean scientists to look at sponges and actually the microbes of the sponges. But I just wanted to show this picture because it was really cool. I haven't much had to worry about ice before when I'm surfacing from a dive. Now moving to Canada, this was in the Arctic. So I was working with a group in the Arctic, and we were surveying the biodiversity there. And again, this was really cool because not a lot of people have been able to dive here, and there's some really cool animals that live here. Like this is an upside down jellyfish. For some reason, it just lives upside down. Okay, so now I've showed you all these pretty pictures, all in the ocean, but why are we even talking about this? Why are we talking about the ocean? Well, as you all probably know and have heard many times, the ocean is stressed. So there's things like ocean warming, ocean acidification, pollution, like plastic pollution, I'm sure you know about, deoxygenation, the ocean's losing oxygen, just to name a few. The list will go on. But I think it's really important to remember, and I always like to start my talks with a reminder that our health and livelihoods up here on the surface is intimately connected to ocean health. Even if you're really far from the ocean, your life is affected by how the ocean is doing. Okay. And a metric that we can use to look at the health of the ocean is biodiversity. So I really like to think that a biodiverse ocean is a healthy ocean. And biodiversity essentially just means the number of different species that live within a system. And that is because each species, no matter how small or how insignificant they may seem, play an important role in the system because it is a system and everything relies on everything else. Okay. But the world is losing biodiversity at an unprecedented rate. And so today I'm going to be talking to you about a group of fish that I am doing some research on. They are called rockfish. So these rockfish are one of the groups that have been declining in the last decades, mainly because of the rise of poorly regulated commercial fisheries alongside climate change. And these fish are very important and pretty cool for a lot of different reasons. First of all, they are culturally important food fish for First Nations here in British Columbia for millennia. They are also a flagship species, which means that their role in the ecosystem basically is vital. And so by protecting rockfish, we actually confer protection to associated habitats and species. And also, they're really cool. They make a lot of sounds. So this one right here that you're looking at is a China rockfish, and they grunt at you underwater. They're like, why are you at my house? And they grunt at you to go away. And they actually can also live to over 200 years old. There's fish in the ocean right now that were born in the 1800s. How crazy is that? So, to address the declines of these rockfish, the Department of Fisheries and Oceans in Canada established a network of rockfish conservation areas. So protected areas like these can be immensely successful in promoting species and ecosystem recovery when placed appropriately. Like those examples that I talked about earlier, Revillagigedo and Cabo Pulmo are two amazing global success stories of marine protected areas. But like all protected areas, we need to monitor them to make sure that they are doing what they're supposed to. So are we seeing rockfish return? Are we protecting the right habitats where the rockfish want to live? Currently, the First Nations on the central coast of British Columbia use fishery-independent methods like scuba diving to monitor sites within their territories, including in these federally placed RCAs. So what you're looking at here is me diving on a reef in the central coast of British Columbia. And diving is a very important tool because we can use it to count fish. We can use it to identify fish. We can make behavioral observations, like some fish are very curious. They love to hang out with you, whereas other fish are very shy and they like to hide. We can also look at habitat complexity. So for example, this place is really cool because there's a lot of really good places for fish to hide, whereas a sandy bottom isn't going to be a great place to protect. We can also look at things like invertebrate cover. So like these plumose anemones, these white anemones. And we can also look at diversity in general, so diversity of the entire system. And all of these parameters are very important when deciding where to put a protected area. Okay, but there are challenges to dive surveys. So first of all, it's very resource and time intensive. Like I said earlier, we're diving in some places that are really, really hard to get to. And in Canada, we often have really cold and very violent weather. And so as divers, we first of all need to be good enough cold water divers that we can handle these harsh conditions. And because we're doing science, we also have to know all of the species there. So not any diver could just come and do these surveys. We're also depth and time limited, meaning that we as divers can only really go to 30 meters for this type of work that we're doing. And rockfish live from the surface to over 1,000 meters deep. So obviously we can't reach all of their habitats. And we're also time limited. So at 30 meters, I actually only have 10 minutes and I quickly have to do all of my work in my survey because we can only have that amount of time because of the nitrogen that's accumulating in our body. So that brings me to what I do for my research, and this is environmental DNA. Environmental DNA is basically genetic material that is found in the environment. So I don't know if you guys know what, you do probably know what DNA is, but it's essentially a molecule that encodes life, and it is in all of our cells. So you know how like we're always shedding skin and hair into our environments? So is everything else. And so essentially what we can do is we can take a bottle of seawater, filter it out all the DNA, extract the DNA, and then ask questions about what's going on in the environment. We can ask questions like what species are here, how many fish are here, or how many of whatever species we're looking at are here, and are there rare or important species here? So this project, this rockfish project, what we decided we wanted to do was we wanted to compare the environmental DNA methods. You can see me here collecting some seawater to our dive survey methods as a tool to assess rockfish biodiversity. So again, like the number of rockfish species that we see in an area. And eDNA is so cool in my opinion because this technique is very useful for community-based monitoring. So anyone, unlike the diving, anyone can go out and grab a bottle of seawater. That isn't very difficult to do. And then communities themselves can be involved in the monitoring. We can, in one day, we could do 4 dives in maybe 2 sites, but we could take 30 samples, eDNA samples from all over. So that is one of the goals of this project, was to try to see if we can make this technique useful for community-based monitoring. So I just wanted to show you, this is where we dove. This is the territory of the Kitasoo First Nation, and it is on the central coast of British Columbia, and they live right in the heart of the Great Bear Rainforest, so that's actually where all the spirit bears live. But this is, all of those pink squares are where we dove. And this is honestly one of the coolest places I've ever been able to dive in my entire life. Okay, so I'm just going to talk about some of the results that we found that I thought were pretty exciting. So what you're seeing here is a classic Venn diagram, and you can see that both methods, so with both our dive surveys and our eDNA, we were able to detect most of the species that we see that are there. So that is really cool. That kind of confirms that, okay, like if we're just trying to look at rockfish biodiversity, eDNA might be a good way to take a snapshot of what's going on a larger spatial scale. With the dive survey, though, we saw 2 species that we didn't detect with our eDNA signal. But with our eDNA, we saw 7 species that we didn't detect as divers. And what I think is cool about that is the fact that the fish that eDNA detected were the ones that were rare or the ones that like to hide a lot. And also we got 4 of those species that are really deep. So their depth range starts at around 40 meters. So they're probably just below us and we didn't see them. And so I think that's pretty cool. Okay, so I just want to end with sort of a reminder of why is this important. It is important to look at the ocean with many different lenses. Like I said earlier, dive surveys are so important and they give us so much information. But eDNA is also a really cool new method. And as our techniques and technologies are getting better and cheaper, it's just going to be a way that we can use on a much larger scale to monitor ocean biodiversity as we continue to go through this biodiversity crisis and try to stop it in its tracks. And so as the oceans continue to change, we need more people like you. Hopefully I inspired one of you guys to become a molecular biology nerd like me, but we need more people like you in sustainable ocean careers. And the exciting thing is we're in a time where there are going to be a lot more sustainable ocean careers because we're all realizing, especially at meetings like this, that we need the ocean to survive. And if we keep on extracting things and not treating it with respect, that's not going to happen. And finally, again, we are all connected to the ocean, and it's so important to remember that. So with that, thank you very much. This is a photo of me with some penguins in Antarctica, and thank you.
Awesome. What a great spot. What a great photo to end, by the way. Neha, if you want to come out of screen share, see us again, have a bit of a conversation, we'd love to have you join. We got a bunch of classes live with us, over dozens of people joining us on YouTube. So if you want to share questions in the YouTube chat, please do. And you were so keen on the mimic octopus, I put it in the StreamYard and YouTube chat if anyone wants to check them out. They're a pretty special creature to see. We're going to head to Ewing, Virginia, Miss Gully's class. If you guys want to come on in first to kick us off with a question, I'm going to come to you guys. We've got our help environmental leaders, Mr. Stillman, Miss Alex, Miss Edie. We got all the teachers today. And so welcome in, everybody. But yes, Miss Gully, I'm going to kick off with you. We'll head back now and come on into Virginia. Unmute your mic. All our teachers can unmute their mics. And you are good to go. Hi.
Hi there. This is Jackson. He has a question.
How do you collect DNA?
How do you collect it? What are you using?
eDNA.
Yeah.
Yeah, that's a great question. So you can use a lot of different things. A lot of times we just use Nalgene water bottles. So we'll just take a water bottle, take a scoop of seawater. What you were seeing in those pictures on my presentation, we were using something called a kangaroo bag. And that's actually the bags that you see at the hospital that connects to your IV, but they're very useful for diving because they're collapsible, because it's hard to bring a big bottle under the water. So we use these collapsible bags and then swing them around and fill them with seawater. And then we'll filter all of that water so that the DNA ends up on a tiny little white filter. And then we extract the DNA from that.
Very, very cool. That was the most vigorous scientific demonstration we've ever had in the history of our broadcast, by the way. So thank you very much for that with the kangaroo bag. We're going to head to London, Ontario to Ms. Trempledger's environmental leaders. And I want to note, you talk about inspiring kids to do amazing stuff. I've never run into a class that has done more since I've started exploring by the seat of your pants than Ms. Trempledger's group. So way to go, environmental leaders. You guys are awesome. And I look forward to your question. Come on in, unmute your mic, and you're good to go. Hey, great tits.
Hello.
Hey.
Hi, I'm Soraya. What is the range that the eDNA can reach in the water? Like, do you know?
Like range, you mean like distance away from the source, or?
Yeah.
That's a very good question. And that's one that scientists are struggling to answer. So eDNA originally started only in 2008 in fresh water. And in fresh water, it's kind of easier to think about how water moves, because in a lake, we do get some movement, but we don't, it's not too hard to model. And a river only moves one way. Whereas in the ocean, we have all of these complex currents. We've got tides, we've got wind at the surface. And so DNA will move a lot in a way that's really, really hard to predict. And so some studies have found that you can detect it up to hundreds of meters away. But then other studies have found too that like a kelp forest ecosystem will keep its eDNA in its little kelp forest. And again, it's because there's not a lot of water moving through there. So that's a really, really good question. And the answer is it depends on the oceanography and the water movement in the area.
But it's still interesting that you said hundreds of meters, because this is something that I've always found really interesting about eDNA, is the thought that you could get a full signal. Oh, there's this Greenland shark and it's on the equator and we never knew that before. And is it just not DNA that's coming all the way down? But hundreds of meters rules that out. Like we don't think that you can get DNA from something that's across an ocean basin. That would never happen. It would disperse in a way that wouldn't give you that signal. So
that would be really rare, yeah.
Yeah. Mr. Stelman's class, Burlington, if you guys want to come in, grade 5s, you're good to go. Miss Alex and Miss Edie, I'm coming to you two next. Hi, guys.
Hi. Like, what's your favorite fish, like type of fish, and why?
Ooh, no pressure.
I love that question. Thank you for asking that. So I think my favorite fish is, it's a really cool fish. I'll talk about my favorite fish in British Columbia, but it is an animal called a grunt sculpin. And Jesse, if you could find a photo and put it in the chat, that would be cool. But the grunt sculpin is so cool because, first of all, they're only this big, and they also grunt underwater. I don't know why a lot of fish grunt, but they're really good at it. But they have evolved so that their head looks like a closed barnacle, and their tail looks like a feeding barnacle. And then they live in old, dead barnacle shells, so they're empty. And then they just pretend to be a barnacle, and they're so good at it.
This is the freakiest thing ever. So I learned a few things in the course of these 4 broadcasts, which I've got to say does not happen very often. I get the chance to work with a lot of oceanographers. I've never heard of this fish, and they're so freaky. Look at this fish. It's
they're so cute, not freaky.
It looks freaky good. It looks like it's like play acting. It's like a magic shellfish, but not a shell. Anyway, that's hilarious. What a fantastic creature. Great answer. That's one of our most unique answers we've ever had to that question ever. So thank you for that. Miss Alex is our incredible crew joining us in Hamilton, grade 6s. If you want to unmute your mic, you just have audio, but we should be able to hear you, and you're good to go, guys. Hey.
Okay.
Yeah.
We have a couple questions. I'll let one student ask, and then I might.
We'll come back around. We should have time for a second round, so you'll be good.
Oh,
yeah.
Okay. So you want to go? Yeah. Okay.
Perfect.
What's the weirdest fish you've ever saw?
The weirdest fish? Oh, I love all these questions, guys. I just get to nerd out about fish. Okay, the weirdest fish, I think, and again, I'm just going to stick with British Columbia because otherwise the world's way too big. But the weirdest fish I've seen in British Columbia is also this really, really cool fish. They're called a ratfish, and they're a type. They're related to sharks, but they're another branch called chimeras. And they actually flap around the ocean. It looks like they're flying, but they're super weird. They've got gold on their bodies, and they've got like they're called ratfish because they've got huge, creepy looking teeth that they use to eat clams in the sand, in the sediment.
So for me, when I see a ratfish, I used to work at the Ripley's Aquarium in Toronto. They had ratfish for a spell, and they look like they've got stitching on them. They look like a Frankenstein monster fish, like something's been put together from other fish. They're really, really unique. And like sharks, like rays that they're related to, they're cartilaginous. So if you feel your nose, feel your ears, that's what their skeleton's made of rather than bone. They're really a quite unique creature. Very cool.
Yeah, and they actually also have a venomous spine on their dorsal fin, and it's venomous enough to kill seals that try to eat them.
Really shocking if you're a seal. I don't think you see that coming when you bite the ratfish. Yeah. Miss Edie's our North Carolina crew. Asheville 5th graders, come on in and take us away. Hey, guys.
Hello.
Hi, Global Explorers.
Hi. My question is, How far can sound travel underneath water?
Ah,
That's a great question. So sound travels, I think it's 4 times faster underwater than on air. So it can travel, or sorry, 4 times farther than it does on air, in air. Honestly, I'm not uh, a sound scientist, but sound does travel very far underwater, and that's why like whales and other animals that vocalize underwater have to use really low frequencies, because those long wavelengths can travel really, really far. And that's why noise pollution is actually a huge issue that's affecting a lot of animals that echolocate. So whales, dolphins, they get really, really messed up by underwater noise.
yeah. We've thank you for that answer. Again, I know it's tricky when we get into the weeds of technical things. We've had a bioacoustician, so a person who studies marine acoustics underwater, Bill Halliday. If you want to check him out on our YouTube channel, some really, really great stuff on the sounds in the Arctic Ocean in particular. My favorite fact about ocean sound is the idea that, and we have proven this is a recorded thing, you can record and hear blue whales across ocean basins. Like they go down to a certain depth where their sound just resonates along the ocean basin. So blue whales can talk to each other across oceans, which is astonishing. One of the coolest facts ever in all of natural history. If we have any YouTube classes, please feel free to share your questions. As usual with this IMPAC5 series, we are like whipping through this Q&A. You guys are great. So I'm going to head back to Virginia Muscully's class. If you guys want to come back in for a second one, come on in, unmute, and you are good to go. Hey guys. Run. No, don't run. Calmly walk.
Hello again.
Hey.
Hey, I'm Mason.
Hi,
Hi,
Mason.
How is the ocean losing oxygen?
Yeah.
Oh, that is a really good question. So there's some fundamental like chemical processes that are happening, but naturally there are a lot of zones in the ocean that are naturally anoxic. So basically there's all these bacteria there that consume all the oxygen. And what's happening now as the oceans warm is that this zone is basically growing because those organisms are just thriving in these new conditions. And so they're eating all this oxygen and they're depleting it faster than it can be brought in from the surface or brought in as like the oceans mix. And so basically it's because as the oceans continue to warm and things continue to change, the layers of the ocean are not as good as mixing. And so as that little anoxic, that layer that's natural grows, it's not getting mixed as well anymore. And so it just keeps growing and growing. And it's a kind of scary thing to think about.
One of the things that you'll see in news sort of about oxygen depletion in the ocean are these dead zones. So specifically the Gulf of Mexico is a place where this seems to happen a lot. And you'll see images like this one where you've got these algae blooms that sort of fill the area and sort of choke out all the other wildlife. And you end up with an area where no fish can survive because there's organisms that have sort of taken over that wouldn't necessarily be doing that were it not for some human impact. So I'm really glad we got that question. Great job, guys. Environmental leaders, back to London. If you guys want to unmute, you're good to go. Hey, guys.
Hi, they dismissed, but I'm still here with a list of questions. So, the question, we have a bunch of vernal pools here and some kettle ponds that were leftovers from the glaciation. Can we do eDNA in our freshwater ponds? And are there things that we need? Like I'm a geographer, so I don't know about these things. So you'll have to fill me in.
Yeah.
Well, I don't know exactly where you're located, but eDNA started in freshwater. And so there's definitely you can definitely do eDNA in freshwater. But it is cool. There's a lot of citizen science programs and environmental education programs that are trying to do this. And so I would look in your area and see if there's any citizen science programs that would support that, because it's so easy. It's a really fun thing to do as a class, to go out, collect the water and filter it. And then you do have to send those filters to a lab for processing, unless you have you need qPCR machines and the molecular things. But the actual process of collecting the water and filtering it, you can definitely do. And then maybe see if there's a lab in the area that you can work with.
Cool. Mr. and Pleasure, actually do follow up with me because I can help you find labs that might be willing to do that. It's tricky offhand really quickly to find eDNA kits that are accessible for classrooms, but I'm sure it exists. And if not, some of the universities in that area would be more than happy to help. So bug me and we'll make that happen. I'm going to head to Mr. Stelman in 2 quick seconds, but we got a question from our German audience today. They were wondering why it might be that you couldn't detect those 2 species using eDNA and what the limits of the technology might be. So any insight on that would be great.
Really amazing question. So one of the limits, one of the facts about eDNA is it really relies on the genetic sequence differences between species to recognize them. And rockfish are really amazing, but they're hard to work with because they are so closely related. So their gene sequences are very, very similar. And so some of the limitations in the entire field is the fact that we first of all have lack of sequence data, because sometimes even the same species will have different sequence if they're from a different area. And so that is just one of the limitations, is like we can only really get to a certain confidence level of identification. The more sequence data we get, the more confident we're going to be. With those diving, with those 2 species that we collected while diving, I honestly have a feeling that they are both so for example, one of them is called the dusky rockfish, and eDNA picked up a species called the northern dusky rockfish. And if you look at them side by side, they look very similar. And so I honestly think that we misidentified one of those species, and that's one of the limitations of dive surveys. So the fun thing about working with multiple monitoring methods is just realizing that each one has its own limitations, and they just work way better together.
Great answer. Very nuanced. I love that. By the way, this is totally unrelated to your topic, but the person who asked that question on YouTube's name was Dr. Barrel Eye. And since I've been showing all these cool pictures of weird fish, I'm going to bring this up for everyone because alongside the mimic octopus, everybody should look up the barrel eye when you're done this program. Because if you want a freaky fish and some of the freakiest evolution ever in the history of the world, this fish is it. Like, that is some amazing deep sea creepy stuff going on. I love it. So thank you for that name and inspiration. Burlington, Mr. Stelman, great job. We got a student like right at the camera. You guys are ready to go. So come on in. Hey.
Hi, my name is Aaliyah. My question is for you is, how long have you been doing this and when did you start?
Thanks, Aaliyah.
So with the eDNA stuff, I started working in a lab. I just was working in a lab in a couple years ago in 2017, 2018 was when I first learned about eDNA and I started working with it. And then since then, it's just really grown a lot. And now I've been seeing it more and more in ocean spaces. And so then when I decided I wanted to go back to school, I decided it's a fast growing field, and we need to make sure that we are careful about using it so we don't make promises that we can't keep. And so the answer to your question is I started working with eDNA about 3 or 4 years. Yeah, I guess 4 years ago. Yeah.
Fantastic. Thanks, Aaliyah. All right, Miss Alex's incredible crew, I'm coming back to you, and then we'll wrap up with Miss Edie in a minute. This has been such a great question period. You guys are awesome today. Miss Alex, come on in. Turn on your mic. Turn on your camera. Whatever you want to turn on is great. Come on in.
We had a question from Kai, and they were wondering if you've ever come across an unidentified species in your diving experiences.
Great question.
Unidentified species. So with my diving experiences, I personally have not come across a new species, but I have worked with some deep sea scientists, I have worked with some deep sea scientists, and then also I've been working with groups where we look at like little worms in the sediment. And recently we found a species that we thought, or everybody's been calling it another species because they look exactly the same, but just because of we tried to detect it using eDNA, and we're like, this should be working, like why aren't we picking it up? And we realized that it looks the exact same, even under a microscope you can't tell them apart, but it's a totally different species. And so now we're like, oh, what do we do about this?
Cool. I love that you mentioned the deep sea. This is something that we get to feature on the broadcast all the time with deep sea researchers. And like the tropical rainforest, every time, and I mean every time, we send scientists down to the deep sea, they find new species. Like every time we look, there's new things there, which is one of the coolest facts in the world. Like if you end up being a deep sea biologist, the world is your oyster. There's like 95% of the deep oceans to discover, and it's just incredible. So I'm really glad we got that question, and I hope our kids take the chance to check it out. We've done like dozens of programs on our YouTube channel. So if you want to go see that as a follow-up, head there right when you're done. Neha, this has been so much fun. We've got time for one more question before we wrap up. So I'm going to head back to Asheville. Miss Edie's group, come on in. Hey.
Hi, my name's Desire. What's the biggest sea creature you've seen underwater, not including sharks or whales?
Not including sharks.
Not
or whales.
including sharks or whales. We're narrowing it down.
Well, that's a really awesome question. Not including sharks or whales. In Canada here, we're really lucky. We have stellar sea lions. And so they're the biggest sea lion in the world. And they are very curious to dive with. And it's really fun diving with the girls because they're very fun and playful, and they come check you out and nibble on your hood, which is sometimes kind of freaky, but they're just trying to feel. But diving with the males is honestly kind of scary because they're massive. They can weigh as much as a car, and they'll just hang beside you upside down, really creepy, and they'll just be making eye contact with you. And I'm always like, oh my God, please don't look at me. But yeah, sea lions would be.
That is like your fifth totally unique answer in the history of Exploring by the Seat of Your Pants, which is hard to pull off. We've done a lot of broadcasts, so thank you for that. We'll look out for those male stellar sea lions when we're off the BC coast next time. This has been so much fun. I want to stress that people want to learn more about the amazing work you do. See some of your incredible photographs. They should head to your website. As I noted at the beginning of this broadcast, this is part of our IMPAC5 series of programs. Check out all the amazing programs being done for IMPAC5 and our two incredible partners, the Canadian Ocean Literacy Coalition and the All Atlantic Blue Schools Network. Neha, you've done one of these before, so you've seen this in action. But what I want to do to wrap up this broadcast, like we do with every single one, is bring in all our classes, say a big thank you and farewell. Miss Gully, Miss Alex, Miss Edie, if you want to unmute your microphones, come on, get those screaming voices ready and join me. You are all in the program. Thank you so much, everybody. Have a wonderful day.